Cell, the unit of life. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. 8.1 What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. In cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement, leading to the electron microscope, revealed all the structural details of the cell. 8.2 Cell theory. In 1838, Matthias Schilden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodor Swan, 1839, a German zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissue, that the presence of a cell wall is a unique character on the plant cells. On the basis of this, Swan proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. Sheldon and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divide and new cells are formed from pre-existing cells, omnicellula, e-cellula. He modified the hypothesis of Sheldon and Swan to give the cell theory a final shape. Cell theory, as understood today, is first, all living organisms are composed of cells and product of cells. Second, all cells arise from pre-existing cells. 8.3 An overview of cell. You have earlier observed cells in an onion peel or human cheeks cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outermost boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material DNA. Cells that have membrane-bound nucleus are called eukaryotic, whereas cells that lack a membrane-bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structure called organelles, like the endoplasmic reticulum (ER), the Golgi complex, lysozyme, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on rough ER. Animal cells contain another non-membrane-bound organelle called centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasma, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell of the egg is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cell. 8.4 The prokaryotic cell are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. Figure 8.2 They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-shaped, rod-like, coccus, spherical, vibrio, coma-shaped, and spirillum, spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in the mycoplasma. The semi-fluid matrix filling the cell is the cytoplasma. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confirms certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor. This plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotes. Have something prokaryote cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially infoldings of cell membrane. 8.4.1 Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three layered, so that the outermost glycocalyx followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelope and the manner in which they respond to the standing procedure developed by Gram-Witch. Those that take up the gram stain are gram positive, and the others that do not are called gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from rusting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of the plasma membrane into the cell. These extensions are in the form of vesicles, tubule, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasma called chromatophores, which contain pigments. Bacterial cell may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside flagella, villi and fimbria are also surface structures of the bacteria but do not play a role in motility. The villi are elongated tubular structures made of special protein. The fimbria are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. 8.4.2 Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer to 20 by 20 nanometer in size and are made of two subunits, 15S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. The ribosomes of a polysome translate the mRNA into a protein. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasma in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasma. Example: phosphate granule, cyanophysin granule, and glycogen granule. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryote includes all the protists, plants, animals, and fungi. In eukaryotic cell, there is an extensive compartmentation of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are differ as the former possess cell walls, plastids, and a large central vacuole, which are absent in animal cells. On the other hand, animal cells have centriole, which are absent in almost all plant cells. Let us now look at an individual cell organelle to understand their structure and function. 8.5.1 Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of the plasma membrane. This study shows that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. Figure 8.4. In addition to phospholipids, membrane also contain cholesterol. Later biochemical investigations clearly revealed that the cell membrane also possesses proteins and carbohydrates. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid mosaic model. Figure 8.4. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important function of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion among the concentration gradient, so that from higher concentration to the lower, water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, so that from lower to the higher concentration. Such a transport is an the energy dependent process in which ATP is utilized and is called active transport. Example, Na plus K plus pump. 8.5.2